everyone. Welcome to another episode of the Points on Product Call. So excited today. My name is Dr. Don D. Nettles. I'm going to be hosting today. Though the picture is Greg and Patty Polka. I'm using their Zoom channel because it's just how this has always been done, and that's pretty amazing. But I'm not an imposter. I am not Greg. I am not Patty. I am Dr. Don D. Nettles. I'm going to be your host today, and we're going to be talking about the new NAD study. And Dr. Brian Dixon, our amazing Senior VP of Product Development, Senior VP of Research and Development, is going to be talking about that and probably taking a pretty deep dive. So really looking forward to that. But before he joins us, I want to kind of tee this up a little bit, because if you're not familiar with NAD, other than we have a product called Protandim NAD Synergizer, NAD has been being talked about in scientific
Dondi, we can't hear you. There you go. You're back.
Back? Okay. Lost me there for a little bit.
Yeah, you just froze for a moment.
Okay. So as I was saying, in 2016, Dr. Yoshinori Oshumi from Japan won the Nobel Prize in Medicine in 2016 for his research on what is called the discoveries of the mechanisms of autophagy. And if you're not familiar with the term autophagy, it's what the whole keto movement is about. It's what the whole intermittent fasting movement is about. When you deprive your body of calories, it needs to get calories from carbohydrates elsewhere. So what does it do? We have a very good backup plan, and the cells actually start to consume their own debris for fuel. We call this autophagy. So that process, when you do a keto diet, when you're depriving yourselves of caloric carbohydrates, or you're depriving yourself of calories as a whole, your body still needs those carbohydrates. It still needs those calories because your cells still need to do their job that you rely on them to do. So the calories have to come from somewhere. The carbohydrates have to come from somewhere. And if you're not getting them through your food, your body has an incredible backup plan called autophagy, where your body the cells will literally consume their own waste, which is called autophagy. Auto means self, phagy means eating. So in the term autophagy, it's literally consuming your own cellular debris. Why that is beneficial is your body will do that to break down fat cells, to use that, break that down into carbohydrates and use that, which is why we lose weight when we go on a keto diet. It's why we lose weight when we do things like time restricted eating, where you only have a certain, 5 hour, 6 hour, 7 hour, 8 hour window where you're eating food. And on the other hours that you're not eating food, your body is still having to do the job. But once you burn through the food you're eating, your body has to kick into that second plan. Well, that's called autophagy. It was considered so noteworthy that the people who do issue the Nobel Prize in Medicine in the year of 2016 thought that was the most profound discovery. And guess what it relates to? NAD and sirtuins. Well, if you've ever heard of sirtuins, maybe you've heard of our Protandim NAD Synergizer, but you didn't know what was at work behind the scenes. That's the sirtuins. That's what is involved in autophagy. So this is a big deal. And I'm waiting for Dr. Brian. When you get on, go ahead. If you're already on, because I can't keep track of everyone. If you're already on, Dr. Brian, go ahead and unmute yourself. Let me know you're here, and I'll go ahead and start teeing you up. But if you're not familiar with autophagy, you're not familiar with NAD, you're not familiar with sirtuins, there's a lot of pretty prestigious scientific researchers out there and doctors that are very aware of it because it relates to longevity, and there's a direct correlation with mitochondria. And if you've obviously heard of our Protandim NRF1 Synergizer, deals directly with mitochondria because mitochondria produces ATP, which is the cellular energy your body needs and the cells need to do their job. So if your cells are trying to consume the debris to have calories, but they run out of energy to do so, they're kind of interrelated. So NAD and NRF1, or more specifically, NAD, your sirtuins and mitochondria, are very, very, very intimately necessary. So they are very good friends and they need to work together. And when they do, you have a compound benefit. So it's critically important. That's why our we went originally from our NRF2 and NRF1, our dual synergizer, to the tri-synergizer when NAD came on board. And you obviously saw the compound effects of that. But while we're still waiting for Dr. Dixon, I want to share with you guys a couple books out there. So as I said, NAD is not just something that LifeVantage is talking about, which in the beginning, NRF2 and NRF1 were something that LifeVantage was pretty much the only people talking about. And then as other people joined us, you there, Amy, as other people saw the merits of it, the word started to get out, researchers and scientists and doctors started to converse with one another, it became a much bigger deal. And NRF2 and NRF1 are now a much bigger deal than when our products first came out because the word is spreading. Well, here's kind of the cool thing. The word on NAD was already out. We just further defined it. We took
the stream
conversation that was already there and we made it a much more compelling discussion by the addition of what we have with our Protandim NAD synergizer. So if you've ever heard of Bulletproof, started out as coffee, Dave Asprey, who actually coined the term biohacking. He was the guy who originated that word. He started a company called Bulletproof, and they were dealing in coffee, specifically coffee that had no mycotoxins or mold in it. Mold in coffee is one of the very common things that comes along with as a byproduct of that. But people think when you boil it, it kills it. Well, sometimes it does, sometimes it doesn't. So he figured, let's just take the mold out altogether. And we have mycotoxin-free coffee, which he called Bulletproof. And then added some MCT oil, added some ghee or some organic butter, blended it up. It's now brain food, and it became a very popular thing. Well, Bulletproof is now not just coffee. It's a whole litany of products, but it's this wellness movement, this biohacking movement that Dave Asprey, the guy that founded that, started. Well, he has a book, and the interesting term or the title of the book is called Superhuman. Now, that is all of our potential. The book is called Superhuman. The subtitle is The Bulletproof Plan to Age Backward and Maybe Even Live Forever. Pretty compelling title. But what he talks about in the book on page 151, he talks about NAD. And he says this: unfortunately, your levels of NAD decline by 50% between birth and age 50, and they continue declining even further after that unless you do something about it. Well, guess what? At LifeVantage with our Protandim NAD Synergizer, we give everyone the ability to do something about it because NAD is pretty dang important, especially when we're talking about life in general, and especially if we want to get into a subcategory of that, longevity, and the longer we live. Well, that's pretty amazing. So this book, Superhuman, talks all about the benefits of NAD. But there's another guy that gets probably most of the press when NAD is discussed, and his name is Dr. David Sinclair. He is an MIT and Harvard researcher. In my opinion, other than Dr. Brian Dixon, he's probably the world's leading authority on NAD and things like that. But in his book, he has a pretty amazing book called Lifespan. So Lifespan, Dr. David Sinclair's book, and the subtitle is Why We Age and Why We Don't Have to. All about NAD. It's all about the NAD pathway. And he's got into some pretty amazing science. I'm going to turn this over and let me text Dr. Dixon to make sure he's understanding that we're supposed to be. Oh, okay. He's joined. He said, Rai just told me he's here. So in this book called Lifespan, Dr. Sinclair talks about two populations of mice. They use mice as a study. Now, none of us on this call are mice, at least that I can see. Some of you don't have your cameras turned on, but I'm pretty sure you're not mice because I don't see Mickey Mouse anywhere in the names here. But so we are mammals. We share that in common with mice, but we're not mice. But the proof of this was pretty amazing. It took two populations, young mice, and deprived them of NAD. And what happened was they aged extremely rapidly. Their brown fur started to turn gray. Their normal, youthful joints started to become arthritic, and they started showing signs of aging because they stopped the NAD cycle. Well, they took another population of mice, which were aging mice, and they turned on, they accelerated the NAD cycle, and they're already gray hair because these were aging mice. They're already arthritic joints. Their hair started to turn brown again, their fur, and their arthritic joints started to heal and become non-arthritic. Well, then they went back to that young group of mice that they deprived NAD, which caused them to age, caused their fur to turn gray, caused their youthful joints to become arthritic, and they turned the NAD back on. And the whole process reversed. So they learned something pretty compelling about this study, is that you can cause mice that are young to age prematurely when you stop NAD. And when you turn it back on, you can reverse the signs of aging. And when you turn on or you accelerate the process of NAD in aging mice, you can actually slow down the effects of aging, in some cases reverse it. Pretty compelling information out there. And this was done prior to us knowing it, prior to the study that Dr. Dixon's going to talk about, and prior to you guys knowing about Protandim NAD Synergizer. So the cool thing is we've got an amazing product, in our opinion, the best product on the market to influence the NAD pathway, but the conversation was already begun. We just joined the conversation and took it up a notch. Or as Emeril Lagasse, bam, we took it up a notch. And that's what we are here to do in many aspects of our life. So consider yourself blessed. And I'm now going to stop talking since Brian Dixon, Dr. Dixon is here. I'm going to turn it over to him to talk about this amazing NAD study we have. So Dr. Dixon, take it away, my friend.
Thanks, Dondi. Gosh, I could listen to you all day. I think you should just keep going with this. But I appreciate you bringing up Sinclair and his book, and a lot of good tactics in there to help us live as healthy as possible. And it's also really interesting you brought up that book, because rumor has it, I might have some inside information, there's another book that's going to be coming out that's really all of the aging experts are getting together, writing different chapters about how we can all age just a little more healthily. And also inside note that you're going to be seeing Protandim NAD Synergizer mentioned in that book as well. So how cool is that? And a lot of that's based on the study that we want to talk about today. So literally hot off of the presses. Let me share my screen. We'll get right into it. So hot off the presses, we can see down here the publication date was March 21st. So I think it came out last Wednesday. We were thinking about scrambling and trying to get this done last week, but we figured, well, let's just take our time and ease into it. So brand new study, hot off the presses, a human clinical study that was centered around our Protandim NAD Synergizer. So we jump right in. You guys know that I like to start with the title. So enhanced trial. So that turns out to be an acronym. So don't ask me what that stands for, but it's pretty clever. It'll be in here somewhere. So the enhanced trial, effects of NAD3 on hallmarks of aging and clinical endpoints of healthy middle-aged adults. So some interesting words in this title, right? Like hallmarks of aging, clinical endpoints, middle-aged adults. This is also interesting that they chose specifically kind of a tough demographic, right? They're not really old, but they're also not young as well. It'd be really easy to look at NAD levels and try to improve them in the elderly because we know they decline so much as we age. So this paper is also important because it's in middle-aged, about 50-year-old adults. And then a subset analysis focused on blood cell NAD concentrations and lipid metabolism. So a subset means that this human clinical study was much larger than what's reported here. And these are just the first findings that have been written up and accepted for publication. And I know that there's going to be at least 2 more studies coming out centered on this enhanced trial specifically. The journal, Physiologia, another great journal, impact factor of about 4.5. So again, a great number, reputable journal. And this one is also publicly available. So if people want to get their hands on this study, it's really easy to do. You guys also know I like to look at the author list. So I see if I recognize anybody. And, here we have Tim Ziegenfuss. I have the hardest time with his name, but Tim Ziegenfuss, or Dr. Tim as we call it for obvious reasons. And then I don't know if anybody might have saw Dr. Lopez speak down in Cancun. He was at an elite academy or elite retreat that was down in Cancun. I couldn't make it, but Hector went down there and it was able to present on a lot of the findings around NAD and its role in human health down there. So really cool. I know them both personally. They're great guys. Study was ultimately conducted at Auburn University and then some other interesting centers. So the Center for Applied Health Sciences. And there's one more that's escaping me right now. But all right, so that's kind of where we're at with this study. Let's kind of dive right in. So again, abstract. You guys remember the different parts of a paper: the abstract, intro, methods, results, discussion, references. So, you start at the abstract, you can get a pretty good take of what's going on. So it starts again with just some methodological work, a little bit of intro, methodology, but then we get down to kind of brass tacks and what they found. So if we look here, right, NAD associated metabolites in blood mononuclear cells. So that's just a specific type of blood cell that we have circulating around. Red blood cells, for example, don't have a nucleus. They don't have any DNA. So we know it's not red blood cells. There are also blood cells that have multiple nuclei as well inside of them, but they're looking at mononuclear cells. And this is important because mononuclear cells have been shown to be the best indicator of what's happening in tissues. They're the most like our own cells. And I should say other cells of our tissues in our bodies. So maybe one little criticism, it was only 28 participants, so it is a relatively small study, but the fact that they were still able to see significant differences in some of these things that they were measuring on a relatively small population is also incredibly encouraging. It's easy to get more statistically significant results when you have more people, because the noise, if you will, the experimental variability actually will get compressed. When it's a smaller population, that variability can be much larger. Then we talk a little bit about what they did. So they measured NAD metabolomics. So what does metabolomics mean? And really what it's referring to is all of that biochemistry that's making up our metabolism. So they looked at specifically NAD metabolites that make up this portion of our metabolism, as well as serum markers of lipid metabolism. So cholesterol, LDL cholesterol, and then what's probably the most clinically relevant is the LDL to HDL cholesterol. And this is also important when you are going to the doctor. Don't necessarily look at one number in isolation, but what does all of that information that they've collected on you tell you? So you might have high LDL cholesterol, but if you also have high HDL cholesterol, the good cholesterol, and that ratio gets back into that healthy range, then there's probably not a lot to worry about. So LDL, HDL ratio is incredibly important for a clinical endpoint. Then what they found is, where is it? These biomarkers significantly decreased with NAD supplementation. So cholesterol went down, LDL cholesterol went down, and the LDL to HDL ratio improved. And then it gives the amounts right after that. But then what is so exciting is that ultimately NAD status was improved based on this supplementation. So kind of jump down, intro. This is a great introduction. There's a lot of background on the aging process and then on the individual nutrients. So if you want to put each of those individual nutrients that make up Protandim NAD Synergizer, some good background there. And then as we kind of get into the last sentence here, right? This is always kind of the experimental question and what they're trying to answer in the study. So the purpose of this current study was to examine how 12 weeks of daily supplementation with a theacrine-based supplement that they've branded NAD3, right, with the ingredients, altered NAD metabolites in those peripheral blood mononuclear cells, as well as the serum lipids. Given the preliminary data discussed above, so if you remember, we covered the first study that these authors had done in cell culture. So because of that preliminary data, they felt they had a good and strong rationale to actually move this into humans. And then the methods, again, you guys have heard me say this, but I don't dwell too much on the methods in case I need to go back and reference something a little bit later. You will see something like this in every human clinical study. This has to be included because there are very strict laws and regulations. When you do a human clinical study, you have to go through a series of events to ultimately get approval to be able to do these studies. And so this lays that out here and really tells me that this is a legal study because researchers have been in trouble. Maybe you guys had heard about what happened at Johns Hopkins, and a couple of subjects in some clinical studies died, and Johns Hopkins got really a pretty bad reputation in terms of doing human clinical studies. And they've really clamped down on making sure that any published study is done in a legal and compliant manner. So it just talks about all the different assays that were used in the experiments. And then we get down to the first bit of data. So here, they're basically giving you the subject demographic data. Okay, so we look at if they're male or female, and then they break down, again, quite common in a human clinical study, they'll break down, so there were 6 males, 9 females in the control group, and then 6 males and 7 females in the treatment group. So this is important because if a study is done right, so you've heard single blind, double blind, triple blind, placebo-controlled studies, right? So when they're blinded, what you'll do is recruit all your subjects, and then you basically give them a study subject number, and then that number gets randomly assigned to a treatment group. So you hope that that randomization process also gives you a good equal distribution of who's in your study. So age, weight, male, female, you hope that those break out relatively equally. And why they've done statistics here, right? Remember, you want a p-value of less than 0.05 to be statistically significant. So the fact that this is over 0.05 and in fact is closer to 0.6 tells us that there's great even distribution here with the age. So the average age is 51 and 52, but statistically the same. And then also looking at BMI, right? That body mass index, the really weight to height ratio, again, isn't significantly different. So they did have a good randomization of the subjects in this group. Table 2 just kind of goes into a little bit more depth about the demographic data of the subject population. So body mass, so really their weight. Again, we see no statistically significant differences. So good distribution again. They looked at blood pressure, so both systolic and diastolic. And interesting to note that the average, everyone had a healthy blood pressure range on average, but a little bit of variability, probably some people a little bit over that healthy range, but the average population, everyone was within the healthy blood pressure ranges. And that's important because if you didn't look at this data and then read that abstract where they saw no changes in blood pressure, you might think it's a negative study. But we're going to talk about this in a minute. But that data is important because they were also evaluating the safety of the supplement as well. So the fact that these individuals had a healthy blood pressure and there was no differences means that we did not increase the blood pressure in individuals. And that's an important thing to point out because teacrine is very similar to caffeine, and we know that caffeine can actually increase blood pressure. So you get that vasoconstriction that occurs. But kind of to give you the conclusion first, they saw no differences, which in this case is a really good thing. So we didn't drive people into high blood pressure range. So then they just jump right into it and they look at that lipid metabolism that's going on. So just like the doctor would measure your lipids, if you went to the doctor's office, they did that same thing here. These empty bars are the control group. So pre is before the study started. So you always go in and do kind of a pre-study blood panel work, right? So pre before the study starts, and then post is after the study has ended. So probably the last study day. No differences in time between the control group. So basically their cholesterol stayed the same throughout the study. But in the NAD treatment groups, you see there's that less than 0.05, a significant reduction in total cholesterol. In triglycerides, we don't see any of the asterisks or statistical denotation here. So even though there's a trend for it to be decreased, it wasn't statistically significant. So we can't put too much stock into that data. HDL cholesterol stayed the same. So because we're lowering total, HDL staying the same, and the LDL cholesterol is also significantly decreased, this is where we also see that really positive improvement in the LDL to HDL markers. So even though we had healthy individuals with relatively healthy BMIs, healthy blood pressure, we were still able to positively affect their lipid profile in these subjects. And I think something we would all be happy to see come back on our blood panels. So now, what about NAD? And this is where I might just deviate a little bit, and I'll pull up some slides and we'll talk a little bit about the science that's going on here. But they have NAD. So this is what we're always talking about. And technically this molecule is called NAD+, but we don't usually say that. NAD, if you remember, here, I'll pull up this slide. So if you remember, food goes into the mitochondria ultimately, and that's where the majority of our food metabolism is occurring. And you can see here in the mitochondria, right? This is kind of the biochemical breakdown. We basically convert all fuels, fats, proteins, and lipids into glucose. Glucose gets converted into another molecule called pyruvate. That's what gets shuttled into the mitochondria, and that's where we generate energy from. And remember, when we're in there, we're breaking bonds of that food ultimately. And as we're breaking those bonds, electrons are released, and they have to be captured by something. And that's something that captures the energy in that electron is that molecule NAD+. Once NAD+ catches that electron, it becomes NADH, and it hands it off to some enzymes inside of the mitochondria that ultimately generate the energy currency ATP. But why is all of that important? Because anything that can be an antioxidant, almost by definition, can also be a radical, and it can also be an oxidant. So for example, we never talk about it with vitamin C, but vitamin C is the reduced form. There actually is, it's called an ascorbyl radical. So vitamin C, once it gives off its first electron in its antioxidant capacity, it actually becomes a free radical itself. And then if it gives off another electron, it gets fully oxidized. So I say all of that because anything that's an antioxidant, right? Anything that can give or receive electrons exists as what they call a couple. So like the LDL to HDL ratio, we want to calculate the ratio of reduced compounds to oxidized compounds. And I know that's really complicated type of science, but we can actually use these redox couples, they're called, to gain some insights into what is happening as we age. So another very common couple or redox couple that we will measure as a surrogate for our, let's say, antioxidant capacity of the cell would be glutathione. So just like vitamin C, glutathione exists completely reduced, right, as an antioxidant. There's a glutathione radical, and then there's fully oxidized glutathione that's called GSSG. So this is what our redox ratio looks like, or think of it as your antioxidant capacity. So when we're young, we have a ratio of about 100 antioxidants for every 1 oxidant that's in the cell. But as we age, look how that just gets completely devastated. And we're talking, 10 to 1. So literally we see about a 10-fold reduction in our antioxidant capacity as we age. So we can look at something similar when we look at the couple of NAD+ to NADH, okay? And that becomes important because this is how the sirtuins were ultimately discovered, right? That enzyme that we're ultimately trying to activate by increasing NAD levels. So remember, caloric restriction was one of the most potent things known to increase sirtuin activity, okay? The reason that happens during caloric restriction is you're not having that food available to be metabolized. So instead of converting NAD plus to NADH, that NAD is just staying as NAD plus, and that ultimately raises the ratio of NAD plus to NADH. So similar to what we're seeing here, but in this case, when NAD levels increase, it activates sirtuins. And that's why that discovery that was ultimately done by Sinclair as a graduate student back at MIT was just so instrumental and profound, that now there's this direct tie between metabolism and longevity. So Dondi, did I just confuse everybody, or is there something I can clarify? But I want to set the stage when we talk about this, the NAD ratio especially, and why that's so valuable in this study.
Well, you didn't confuse me, but I think I'm biased because I devoured this kind of information. So one of the things that I know, it's probably it's obviously a very complex subject, but I don't know, I just put it out there. Are you guys confused? What would you guys like clarity on? Because I'm eating this stuff up and I'm waiting for you to talk about the ratio.
Silence is good, I guess, in this case. But yeah, if you have a question, throw it in the chat. Maybe Dondi, I could trouble you to keep an eye on that. Just kind of chime in. Okay,
hey, Dr. Dixon.
Yeah, please.
This is Kevin POTUS.
Hey, Kevin, and please call me Brian, please.
Okay, thank you. So if you, in common terms, like to speak to a layperson, like just in general terms, what is this telling you?
It's telling me that the study ultimately is telling me that we can increase NAD levels. So that's the simple thing I want you to remember. Now, I'm going into some of that minutia of the science, maybe just as background for you, Kevin, and the others. But you don't need to know all of that science. Again, it's not my goal to turn you all into little scientists, but just really how we can assimilate a scientific paper, pull out kind of the important things that we need. But I would just keep it simple and just say we can increase NAD levels. And that's why this is so profound, because I don't have to put you on the worst diet you would ever be on in your life and restrict your calories by 60% to see the same change in NAD levels that we see by a supplemental application that's designed around activating your body's ability to make its own NAD. I hope that helps. I hope that answered your question, but thank you.
Thank you.
All right, let's jump back over to the study. Oh, yeah, we're coming back to that pathway, don't worry. Okay, so now we're coming back to NAD. So now what they're measuring are the two populations, the fully oxidized and the fully reduced. So the fully oxidized NAD and the fully reduced NADH. Okay, so this is coming from metabolism, and this is basically it's been consumed up. Okay, then they see a trend up in NAD, but it's not statistically significant. They see a trend down in NADH, not significant. But where it does become significant is when they look at the ratio of that oxidized NAD plus to reduced NAD, they see a significant increase in that ratio. And it turns out, and why this is so important, is it's not just enough to increase total NAD plus and NADH ratios. You have to have that ratio change, right? Improve, so there's more NAD plus around than NADH to actually get the activation. If you increase the total levels the same, that ratio wouldn't change and you would not see activation of these sirtuin proteins. So while we've used pretty simple words and just talked about increasing NAD levels, it's actually the ratio that is the most important thing to get that sirtuin activation, okay? So then they measured, this turns out to be a metabolite in the NAD biosynthetic process, and a pretty clever approach. And I'm glad that they threw that endpoint in there, because if we go back to the NAD biosynthetic pathway, remember, there are two ways that we can make NAD. We can make it from scratch from amino acids like tryptophan, right? So we can make NAD from scratch, or we can recycle the NAD that's been used up. And this is called the salvage pathway. And it turns out that this almost adds nothing to the total NAD concentrations. And it's the salvage pathway that's the most important. So the reason they measure that metabolite, that NAAD, was to get an idea, were they getting activation from making NAD from scratch, or the de novo pathway, or were they activating the salvage pathway? So they looked at that metabolite as an intermediate, and they did not see any change, with the conclusion that it had to be the de novo path, or the salvage pathway, excuse me, that they were actually activating. Okay, come back over to the paper. So here again, they measure that metabolite, and if anything, they see it trending down, not statistically significant. So really, there's nothing happening on that from scratch pathway. It's all happening through that important salvage pathway. And then they just threw this data in to look at the individual responses of the NAD to NADH ratio. So the conclusion, again, it's fine to just say we are increasing NAD levels because of this data here. Now, something that I think is also incredible with this study is they looked at the safety of taking this product. And to be quite candid, we might have had a lot to do with that. You know, I've heard whispers out in the field that individuals might have some issues when they start taking NAD. So they did an incredibly extensive safety profile and looked at all of these different things, right? White blood cells, red blood cells, hemoglobin, right? All of the different blood cells. And again, if we look at the comparisons, we basically see no changes. There's a couple, you know, that kind of pop out over time. So here there was a slight increase in red blood cells, but for the most part, really nothing changed. There was a slight increase in hematocrit, usually a good thing, but of course, when it gets too high, that can also be problematic, but still within healthy ranges. Again, just looking at more blood cells and then looking at glucose, right? Resting glucose levels, again, all in the healthy range, no significant changes. Blood urea nitrogen, a measure of kind of kidney health. Creatinine, also a measure of kidney health. The ratio, right? Sodium, potassium, chloride, calcium. So an incredibly extensive battery of a safety profile. And again, almost nothing had changed. So now we have scientific proof in a clinical setting where patients were monitored incredibly closely for safety, showing that this supplement is completely safe and very well tolerated over the 12-week study time. And that's really kind of the gist of the paper. So remember what they've been able to show. They've been able to show an increase in NAD levels and improvements of lipid blood chemistry. And then again, we get into the discussion, and this is usually where I'll spend the most time with the paper. Remember, the point of the discussion is to put these findings into the context of the larger scientific context. And I think, especially in these first two paragraphs, there's some really profound things going on. So starting here, significant interactions, right? P less than 0.05 were observed for total and LDL cholesterol, where values significantly decreased with teacrine-based supplementation. Additionally, a significant interaction was observed for that NAD to NADH ratio, where values trended downward from pre to post. So those were those levels that we saw that weren't significant, but they were increased versus the control by the end, okay? So that placebo group. These data collectively demonstrate that teacrine-based supplements can favorably alter biomarkers of lipid metabolism and cellular NAD status. So that really kind of sums everything up right there. This is another good paragraph here, as with our previous report. So that's the initial cell study that they did. So as with the previous report showing 8 weeks of teacrine supplementation reduced serum total and LDL cholesterol concentrations, I'm sorry, this came not from the cell culture study, but another human study that they had done. This current study demonstrates similar findings over a 12-week period, except with a much smaller dose of teacrine as part of this core complex. The physiological effects of supplementation could be due to multiple mechanisms. So now they've gone into the science to figure out how could this be working. So they know the input, they know the output, and now they're looking to link how this actually could be working. And so you'll see they talk about this adenosine receptor. And that's important because we know caffeine is binding to the adenosine receptor. And because caffeine and teacrine are structurally so similar that they are also binding to the same receptor. So what's been shown, right? So we know teacrine can bind to the adenosine receptor, but then in mice, you can do some genetic studies. And when they knocked out that receptor genetically in mice, they got high cholesterol. So a good, pretty strong rationale that teacrine, by binding to the adenosine receptor, is somehow activating healthy lipid metabolism, probably mostly in the liver. So it just raises the question that supplementation enhances that signaling, right? The adenosine signaling in the liver, which in turn lowers cholesterol biosynthesis. So you're actually inhibiting the liver from making cholesterol. So there's kind of that scientific rationale of how this could be working. It's not necessarily pie in the sky. And then they do it with each of the ingredients. So wasabi, what's known, how they're working. The active ingredients in wasabi are known as isothiocyanates. So these are very potent activating compounds. And they go on to kind of put that into perspective, and especially in the context of inflammation as well. But again, just brass tacks, if we come to the last section here, the conclusion, right? These data continue to demonstrate that a teacrine and isothiocyanate, right, the wasabi-based supplement, can positively alter molecular markers related to lipid metabolism, cardiometabolic health, and hallmarks of aging, okay? Moreover, these are the first human data to suggest a teacrine and isothiocyanate-based supplement can positively affect NAD metabolome, so those different metabolites that they measured in human blood cells. And given the limited data in this area, more research is warranted. You know, I always laugh when I see these studies because I already know they're writing up the additional papers. So they've already done the research and they're in the process of writing it up, but more research is warranted. And the next two studies that are coming out are just going to build off of this foundation. So it's going to be pretty exciting next couple of months as it relates to Protandim NAD Synergizer. So that's really the gist of the paper. So now we have human clinical data showing clinically that we can increase NAD levels and we can positively affect lipid metabolism in humans with our supplement. Incredibly powerful. And they really, I talked to both Tim and Hector about this, but they demonstrate that, it says it here, the first human study, or the first human data to suggest a teacrine-based supplement can positively affect NAD. They purposely soft pedaled this. It's quite common to do in a scientific paper, but really this is the first human data that has been shown using any type of supplement, whether it's a precursor or an activator, to actually increase NAD concentrations in human beings. So that gives us right there also a competitive edge, because there are competitors out there that are utilizing a different strategy. But this is the first paper to show us that in the absence of any significant type of precursor, through this activation approach, we can trigger the body to make more NAD. And as a result, we get those kind of downstream effects, that being that lowering total cholesterol and improving that LDL to HDL cholesterol. So part of the first study that's ever been done in humans to show that we can positively affect NAD status. With that, Dondi, maybe I'll hand it back over to you. And I don't know if you saw maybe some questions come through or anything else we want to discuss related to the paper.
Yeah, let me open up the chat. And while I'm looking through the chat, people are just really excited. This is absolutely amazing. Human data, so exciting. Yeah, so great feedback. And interestingly, so when we released this product a little over almost 3 years ago, there was, as I kind of mentioned as I was teeing you up, Dr. Dixon, is that there's information that was already out there, and something specific in Dave Asprey's book, Superhuman, on page, at the bottom of page 152, top of page 153, he talks about the ratio of the oxidized NAD to the NADH as being normally between 700 to 1. And then he says that most healthy people have this proper ratio in their cells until around the age of 40, when the NAD plus levels begin to decline. He says it's no big surprise that this coincides with increased oxidative stress and rapid cellular aging. Athletes are usually the first ones to notice an NAD decline when they suddenly can't bring it the way they used to, despite keeping up with their usual training regimen. And then other normal people, in quotes, usually don't notice this, anything has changed until another decade or so has passed. So when people that are athletes, especially elite athletes, they're very in tune with their bodies. They're typically have incredible workout routines, very good cardiovascular fitness, good nutrition programs. They start to notice it pretty rapidly, because the more in tune they are with their body, when this NAD ratio starts to go down, they start to notice it. And your average person who maybe isn't as cardiovascular fit, maybe isn't paying as close attention to their nutrition, maybe doesn't have the extreme physical fitness protocols as elite athletes, they notice it about a decade later, but they still notice it when we have, it says between birth and the age 50, we have a 50% decline in NAD. I'm super excited that this study talked about that ratio, because when we've got valid information, and Dave Asprey, I'm not saying, I mean, he's the guy who coined the term biohacker. He's a pretty influential man in the biohacking movement, and he's very popular in what he does. When he mentions this, and then we now have this study that was published about a week ago to corroborate that ratio, I think it's an incredible arrow in our quiver that we can use information that's already been out there to substantiate and validate what we now have on humans that's published, to really just validate the story that we have now. So great supportive information.
Yeah, it is a wonderful scientific story. I mean, that whole notion of caloric restriction and improving NAD levels was really, well, I should say caloric restriction extending life was discovered in 1935, but it wasn't until Sinclair and his graduate mentor, Leonard Garante, actually did some molecular studies that they were able to link that caloric restriction, increasing NAD levels, activating sirtuins to get that increase in longevity and the improvements in those quote unquote aging markers that we see throughout the course of our lives. Those have been followed up with, numerous experiments in different experimental models, right? Everything from fruit flies to microscopic nematode worms to mice to rats to monkeys. And now it's finally made its way back into the human literature. And now, based on all of that previous research, you would expect that increasing NAD levels in humans would be equally beneficial to health. And now we're building on that story in humans with the Protandim NAD Synergizer.
Yeah, absolutely. And in that book, Superhuman, he does mention one of the products by name that we probably have talked about before. But now when you're talking about chapters coming out that are going to mention our product, Protandim NAD Synergizer specifically, it's going to be pretty exciting in the future of what we can say and what kind of changes we can help people get to. Awesome.
Absolutely. Stuff. Cool. Anything else I can help clarify for anybody? I hope this is beneficial. I know we do kind of deviate into deep science here sometimes on these studies, but it's just more information that you all can go out and talk about. And if someone says, do you have studies to prove it? Now all you have to do is email them or print them out a copy of the publication. So just hopefully making your jobs easier as well.
Ryan, one question, this is Jen Dykstra in Sioux Falls, and one question I guess I have that I always run into is what we can and can't say, or what we should or shouldn't say, as it relates to like medical grade or how to overcome that, especially when it comes to NAD. There seems to be a lot of conversation around that, but no real answers in terms of like what you can find even from other competitors, if you will.
Yeah, Jen, forgive me. I'm not sure I exactly understand the question. Is it how can we talk about these things, or?
Yeah, so oftentimes I get the question, how does your NAD compare to a medical grade NAD? And I always say that it's not medical grade because we are using plant-based products and it goes through our everything that we do from that perspective. But how do you overcome NAD, what we have versus medical grade, and explain that in a normal manner, if you will, non-scientific?
Yeah, I see where you're going. It's really, in my mind, a little bit of a funny conversation. I might sound flippant when I say this, but there is no such thing as medical grade. There is no certification there, right? The FDA, the FTC, there's no regulatory body that approves a supplement to be quote unquote medical grade. I hear quite often too, are your supplements FDA approved? Well, the FDA never approves a supplement. So I think that lexicon kind of makes it into the consumer base for reasons I have no idea. I think people think it sounds sexy and they stick with those words, but it's kind of an untrue premise to begin with, because there is no such thing as a medical grade type of supplement. And so maybe what I would recommend in that case is, try to figure out more and ask questions like, well, what do you mean medical grade? And can you give me some examples? And then from there, we can start to maybe get at the mechanistic approaches to what these supplements are doing. I'm personally not a believer that NAD infusions actually do anything, if that's what they mean by medical grade. I think once that NAD gets put into the bloodstream, it gets destroyed almost immediately. That's why you can't take it in a supplement and appreciably raise your circulating levels. So I would just dive into it and ask them what an example might be, and then we can get at it from a scientific perspective. Are they still harnessing the precursor approach? Are they just infusing or supplementing with just straight NAD, which also doesn't work? But now we have this scientific proof that through our activation strategy, we can absolutely increase NAD levels in humans. And I would, right now you have the best ammunition, because you could say, well, ours has been clinically studied, has yours? Because to my knowledge, to the knowledge of the authors, this is the first paper in humans utilizing a nutritional supplement that increases NAD levels. So right off the bat that that's kind of your ace in the hole, and there is no comeback to that right now. I hope that helps, Jen.
Such a great point, Dr. Brian, because when people talk about medical grade, I think, well, number one, medical grade is a great marketing term, but when people are getting infusions in a quote unquote medical clinic or a multidisciplinary clinic, maybe the inference is, or the assumption is, that somehow medical grade because some kind of a medical employee or medical healthcare provider is actually administering it, doesn't make it medical grade. But to your point just now about in the conclusions portion of this study, where it says these are the first human data, meaning all the popularity, all the people that are participating in that, including Dave Asprey in his book and Dr. Sinclair in his book, this was all non-substantiated information, because this is the first clinical human data to actually be published. So that is great. That is our ace in the hole. So, well, here, according to this is the first data published. The other is just conjecture and hearsay and anecdotal stories.
That is such a great point, Dr. Nettles, because like what Dave Asprey was saying, even what Sinclair is saying in his book and his approach is like, Sinclair is a big fan of one of those intermediates, NMN, yep, in that pathway to try to overcome the rate limiting enzyme there, right, to increase NAD levels. That's all based on experiments that have been done in different experimental models, right? So different animals. So it was all based on the scientific theory. And now we're able to show that that theory can actually be realized and that we can affect somebody's NAD status with a nutritional supplement.
Absolutely. Brings me back to that Harvard bar scene in Good Will Hunting. It was like, how you like me now?
How about them apples? Yeah, wonderful. Well, Don D, thanks for hosting. And I really appreciate you putting everything into context, talking about Sinclair in his book and Asbury in his book. And it just shows the power of what we all have in our hands, when these thought leaders in the aging field are so intrigued with increasing NAD levels and its potential for human health. I appreciate you taking the time to do that, Dr. Nettles.
Oh, absolutely. There is actually a great question that just came up in the chat, and it's just asking, why does the study refer to the product as NAD3 versus Protandim NAD Synergizer? Why is the Protandim name not in there?
Well, a couple of reasons. They have just branded that core blend. So it's just nothing more than marketing of that blend. And quite frankly, I'm happy that they didn't mention the product by name, because some of the papers that we probably won't be able to cover are the products that mention, let's say, Protandim NRF2 Synergizer by name, and then talking about its effects on cancer or some of these other things. So the fact that it doesn't call out our product specifically, I actually view as a strength and not a weakness. And we can more easily talk about the benefits of the product.
Yeah, and no direct inference or potential liability of violating FDA protocols when we're talking about that. Our direct product name, finished product name in association with any kind of disease, again, makes a point. But if we talk about, we actually have the ingredients of our product, and it mentions the ingredients in this study, there could be a great correlation there without having to use our Protandim NAD Synergizer name.
Yeah, and all of the endpoints that they measured in this study, right? If we go back through those tables and figures, nothing was decreases your risk of heart attack or decreases your risk of a stroke or anything. But what they've done is looked at markers of cardiovascular health that are a risk factor. So talking about risk factors and improvement isn't necessarily an issue when we're not talking about disease. But if I said, I have a product that can improve the LDL to HDL cholesterol, I think everybody's mind goes right to cardiovascular health, but we're not saying it. So that's where we can say, it improves markers of lipid metabolism in our blood. I mean, how great is that? And, NAD, improving NAD concentrations, not a medical claim again. So there's nothing in this study that I see as a cause of concern, and we can talk directly at it.
Yeah, to that point, I was really surprised at how extensive the blood markers was. And they talk about, they obviously measuring blood markers that affiliate with cardiovascular health, that affiliate with kidney health, that affiliate with liver health, and that all of them are incredible. It was much more of an extensive list than I was expecting when I first saw this study this morning, but all great.
Yep, and it should give us all peace of mind. The product's completely safe to take. Absolutely.
Yes. Absolutely. Well, thank you so much, Dr. Brian. As always, you just bring the goods, and thank you everybody for taking the time out of your busy schedules to tune into this, to learn more about an incredible product, an incredible study that validates this, and really the position that we hold as the premier position in the ability to not only influence people's general overall health and wellness, but do it at the cellular level in ways that no other products out there or no other company out there can. We are super, super excited to be literally position one in all of those. And but as is the case, when you're position one, you've always got people chasing you and trying to tear you down, which is to be expected in this capacity. But we have incredible documentation now with this study that just really puts us the place that we have always known ourselves to be as the results, or as it relates to our competition, but all of you, to how truly blessed we are to be a part of LifeVantage at this time when we're really changing the world. So thank you guys all so much for attending. Thank you, Dr. Brian. Thank you, Fry. Thank you, Patty and Greg, for letting us use your Zoom room. It's an incredible honor to be here to represent you all.
Yeah, thank you, everybody. Thank you, Dr. Nettles.
Absolutely. Take care, everybody. See you next week.